Welcome to another episode from the Veterinary Medicine Digest. We are standing on the precipice of a neurological frontier where the smallest errors in a biological blueprint can lead to a catastrophic collapse of the most complex system known to nature, the brain. Today we are diving deep into a mystery that spans species, from the quiet research laboratories of mouse models to the clinical halls of veterinary hospitals, and ultimately to the bedsides of human infants fighting for their lives. We are talking about the central nervous system and the protective insulation that allows it to function, a substance known as myelin. But more specifically, we are unraveling a scandal of genetic proportions, a single letter change in a code that should have been perfect. This is the story of a novel canine model that has finally allowed scientists to differentiate the maturation of cells in two of the most devastating forms of a condition known as Pelizaeus Merzbacher disease. To understand the tragedy of these cases, we must first understand the miraculous complexity of myelin. In the central nervous system of vertebrates, the speed of nerve conduction is not merely a matter of proximity. It is a matter of insulation. The axons, those long, slender projections of nerve cells, are wrapped in multiple layers of a lipid-rich membrane provided by specialized cells called oligodendrocytes. This material, myelin, consists of approximately 70% lipid and 30% protein. It is the biological equivalent of high-speed fiber optic casing. Without it, the electrical impulses required for movement, thought, and even breathing would slow to a crawl or dissipate entirely. For decades, scientists believed the protein composition of this membrane was simple, dominated by a few major players. However, modern proteomic research, utilizing gel-free and label-free mass spectrometry, has revealed a dynamic range of over 4 orders of magnitude in protein abundance within the central nervous system myelin proteome. At the heart of this proteome lies a protein called proteolipid protein 1 and its smaller splice isoform known as delta marker 20. Proteolipid protein 1 is a tetraspan membrane protein, meaning it weaves through the cell membrane 4 times, and it is arguably the most dominant protein in the central nervous system myelin, constituting approximately 38% of the total myelin protein. Its role is critical. It acts as a biological glue, stabilizing the compact layers of myelin by securing the intraperiod line, the tiny 2-nanometer space between the extracellular surfaces of the oligodendrocyte process as it wraps around the axon. Next in line is myelin basic protein, which makes up about 30% of the total protein and is responsible for compacting the cytoplasmic surfaces of the membrane. When these proteins are absent or mutated, the results are nothing short of devastating. In human medicine, this manifests as Pelizaeus Merzbacher disease, a X-linked hypomyelinating leukodystrophy. It is a spectrum of disorder, ranging from the classic form, where children might live into their 2nd to 6th decade with significant motor deficits, to the Connatal form, the most severe version, where symptoms appear in infancy and are often fatal within the first 10 years of life. The scientific community has long struggled with a scandal of missing information. Because Pelizaeus Merzbacher disease is rare, there's a profound scarcity of human tissue available for the kind of deep histological and molecular scrutiny required to understand why some mutations cause mild disease and others lead to rapid death. For years, we have relied on rodent models like the Gimpy mouse and the myelin-deficient rat. While these models have provided the foundation of our knowledge, they are limited. A mouse brain does not have the neuroanatomical complexity of a human brain, and these small animals often die too quickly to allow for a study of the disease's natural progression over time. We have even looked to non-human primates, specifically rhesus macaques, where a spontaneous missense variant in exon 5 of the proteolipid protein 1 gene was found to cause intention tremors and nystagmus, mirroring the human condition. But the most recent breakthrough, the subject of our deep dive today, comes from a surprising source, the English Cocker Spaniel. Imagine a litter of 5 puppies born into a world that should have been full of play and growth, but at just 2 weeks of age, a shadow falls over 3 of them. They begin to exhibit generalized tremors involving the head, the body, and all 4 limbs. These tremors stop only when the puppies are at rest. The most severely affected was a male pup, who was notably smaller than his sisters, and whose tremors were so profound they hindered his every move. Veterinary neurologists at the University of Glasgow conducted a rigorous examination, finding a hypermetric hopping response and a mild delay in postural reactions. But the most chilling finding came from the brainstem auditory evoked responses. The interpeak latencies were nearly double those of a healthy pup, a clear indication that the electrical signals in his brain were struggling to navigate a landscape devoid of proper insulation. While his sisters showed a transient tremor that resolved as they aged, the male puppy's condition deteriorated. He became non-ambulatory and was suspected to be blind, failing to track objects in his visual field. At 5 weeks old, the decision was made to humanely euthanize him, an act that allowed for a post-mortem investigation that would peel back the layers of this genetic mystery. When the investigators opened the skull, the macroscopic findings were striking. There was a complete lack of distinction between the gray matter and the white matter in the brain. In a healthy brain, the white matter is vibrant, a testament to the dense myelination within. Here, it was gray and gelatinous. Histopathology revealed a profound paucity of myelin throughout the brain and the spinal cord, while the peripheral nerves remained perfectly normal. This was a disease strictly of the central nervous system. Using immunohistochemistry for cyclic nucleotide phosphodiesterase, an enzyme that should be abundant in oligodendrocytes, the researchers saw almost nothing in the spinal cord. The white matter was a ghost town. Semi-quantitative assessments of markers like oligodendrocyte lineage marker 2 and the proliferation marker Kiel67 showed a massive reduction in the number of positive cells compared to age-matched controls. But the real scrutiny began when they looked closer, using the incredible power of electron microscopy. In a normal pup, you would see axons wrapped in thick, dark rings of compact myelin. In this Cocker Spaniel, those rings were gone. There was a near-total absence of compact myelin, with only the rarest of axons possessing a thin, poorly compacted sheath that lacked the necessary intraperiod lines. Even more interesting was the state of the oligodendrocytes themselves. In the previously studied shaking pup model, which is a Welsh Springer Spaniel with a different mutation in the same gene, there are many more mature oligodendrocytes that manage to produce a success of sorts, a delayed myelination that eventually covers almost all axons by 2 years of age. But in this Cocker Spaniel, the majority of the glial cells were stuck in an immature cellular morphology. They were type 1 oligodendrocytes with large pale nuclei and limited cytoplasm, cells that were trying to become something they couldn't quite reach. The rare cells that did reach a more mature state had something else. Their rough endoplasmic reticulum was massively distended. This distension of the rough endoplasmic reticulum is a major clue in our genetic investigation. It suggests a toxic gain-of-function scenario. You see, the genetic analysis of this pup identified a hemizygous missense variant in the proteolipid protein 1 gene, a thymine to adenine substitution at nucleotide 92. This change resulted in a leucine being replaced by a polar glutamine at amino acid position 31. This wasn't just any position. Position 31 is located within the very first transmembrane domain of the protein. In the world of protein folding, geometry is destiny. Placing a hydrophobic leucine with polar glutamine in a region meant to sit inside a lipid membrane is like trying to force a square peg into a round, oily hole. The protein cannot fold correctly. It gets stuck in the quality control center of the cell, the rough endoplasmic reticulum, causing that massive distension as the cell tries and fails to export the misfolded product. To understand why this specific mutation is so significant, we have to look back at the older models. The shaking pup model, which scientists have scrutinized for over 30 years, carries a histidine to proline substitution at position 37. That mutation affects the very first extracellular amino acid just after the transmembrane domain. It causes a classical form of the disease, one where the cells are delayed and stressed, but many eventually overcome the block and produce thin layers of myelin. But our Cocker Spaniel puppy represents something far more sinister. The Connatal form of Pelizaeus Merzbacher disease, the PL31Q mutation, causes a developmental arrest. It doesn't just slow the cells down, it stops them at the oligodendrocyte progenitor cell stage. This is a critical distinction that has been mirrored in human research using induced pluripotent stem cells. Scientists in that field generated panels of stem cells from 12 different individuals with various proteolipid protein 1 mutations. They discovered that the mutations fall into distinct classes of defects. Some individuals completely fail to generate oligodendrocytes. Others produce cells that are positive for early markers but fail to produce the actual proteolipid protein. And a third group, like our Cocker Spaniel, shows a perinuclear retention of the protein. where the proteolipid protein is made but gets trapped around the nucleus in the endoplasmic reticulum. Interestingly, the induced pluripotent stem cell research showed that the most severe connatal phenotypes were often associated with a significant depletion of those early progenitor cells. When we scrutinize the Cocker Spaniel data alongside these human findings, a pattern emerges. The PL31Q variant in the dog caused an accumulation of immature oligodendrocyte lineage cells, confirmed by an increase in the early marker known as NERV, glial antigen 2, and a decrease in mature markers like oligodendrocyte lineage marker 2, cyclic nucleotide phosphodiesterase, and sirtuin-2. Sirtuin-2 is particularly fascinating. It is an enzyme found in the non-compact compartments of myelin and is a potential sensor of energy status. In these puppies, the normal movement of sirtuin-2 from the nucleus to the cytoplasm was disrupted, suggesting a total failure of the pathways needed for the cell to remodel its skeleton and begin wrapping axons. Now, why does this matter for the future of medicine? If the problem is a developmental block, then a recovery like the one seen in the classical shaking pup is impossible without intervention. In the classical model, the spinal cord eventually recovers because more oligodendrocytes differentiate and survive over time. But in the connatal model, the cells are stuck. Any therapeutic strategy designed to treat this severe form of the disease must be initiated extremely early in life and must focus specifically on stimulating the progression of these progenitor cells. The induced pluripotent stem cell research offered a glimmer of hope by testing small molecules that modulate the endoplasmic reticulum stress response. They found that for certain mutations, specifically those involving gene duplications or certain transmembrane substitutions, drugs like guanabens, which prolongs the stress response to allow more time for protein refolding, could actually restore some of the cell's morphology and allow them to start tracking neurons again. However, for other point mutations, these drugs were less effective, suggesting that the protein might be so badly damaged it is refractory to refolding. The discovery of the English Cocker Spaniel puppy with the PL31Q variant is a game changer. It provides the first naturally occurring large animal model of the most severe connatal form of Pelizaeus Merzbacher disease. It allows us to scrutinize the natural history of the disease in a way that petri dishes and mouse models never could. For instance, it showed that despite the total lack of myelin, the axons in the puppy remained remarkably well preserved at 5 weeks of age. There was no evidence of massive axon death or secondary inflammation, which contrasts with some other models like the rhesus macaque or certain mouse mutants. This tells us that there is a window of opportunity, a period where the wiring of the brain is still intact but simply waiting for its insulation. But we must also scrutinize the biological costs. The proteolipid protein 1 gene is highly conserved, meaning its code is almost identical across humans, mice, rats, and dogs. This conservation is a double-edged sword. It allows us to use these animal models as proxies for human suffering, but it also means that the scandal of a single genetic error is universal. When a leucine at position 31 is changed, the system fails, regardless of whether that system is a human child or a spaniel puppy. Bioinformatics tools like SIFT, which stands for Sort Intolerant from Tolerant, and MutPred2 use sequence homology to predict these disasters before they happen. They calculated that the PL31Q substitution in the Cocker Spaniel had a probability of being deleterious that was essentially certain. These tools have become essential in our era of personalized medicine, helping us prioritize which of the thousands of variants in a personal genome might actually be a death sentence. As we wrap up this deep dive, we are left with a profound sense of the precariousness of life at the molecular level. We have seen how a deep profile of the myelin proteome has expanded our understanding of the white matter from a simple grease to a complex, multi-billion-dollar biological industry. We have seen how the induced pluripotent stem cell models have categorized the failures of that industry into failure to produce, failure to export, and failure to mature. And we have seen how a single puppy in Glasgow has provided the evidence to differentiate those failures in the most severe cases of connatal disease. The English Cocker Spaniel PL31Q model is not just a veterinary curiosity. It is a translational bridge, a living testament to the shared biological heritage of all mammals, and a beacon for the development of early life interventions that might one day allow a child or a puppy to finally stand and run. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging.